This is a safe, productive packing house. These people work hard, they care about each other, and they care about their customers. They're well trained not only on how they do their job, but also about food safety. They believe the safety of this fruit is in their good hands, but not completely. There's an invisible world that can harm fruit. So they've developed a plan using science, data analysis, and experience that tells them how they're doing in guarding against this threat. It's called environmental monitoring. The packing house is an environment of people, equipment, and products. We monitor this environment to make sure it's safe and productive. Environmental monitoring is a verification tool. It verifies or proves that our good efforts really do pay off. Testing our environment tells us if our cleaning program is working. We're better able to identify areas that require special maintenance procedures, and we're able to detect and prevent pathogen contamination. Every packing house has an environmental monitoring plan, and every plan is different depending upon the age, technology, product, and even climate of the plant. But while there are differences, packing houses share many environmental monitoring principles that I'm going to share with you. You'll learn about the different types of sampling procedures. We'll show you how to identify sampling sites and how frequently those sites are tested. You'll see how samples are properly taken, and you'll learn what to do with them when they're ready for analysis. Environmental monitoring reveals the invisible world, bacteria for example, but it begins with what you see with your eyes. Look around your plant. There's standing water that can't be ignored. A glove needs to be disposed of. Leaves and twigs await their final journey out of the plant. Look for opportunities to improve food safety. Watch the processes. Make sure you know how the sanitation crew does their job. Environmental monitoring is detective work. Constant investigation of your environment will help you search for and prevent suspected contaminants. Let me show you how we are able to investigate the invisible world. Environmental monitoring uses sterile sponges, swabs, gloves, Petri film, and other tools to capture and analyze samples on site or through accredited testing labs. When an environmental monitoring plan is first created, a map and master list of every sampling site in the facility is made. Then, a lot of tests are done throughout the plant. These initial tests give us a baseline, allowing accurate tracking of our cleaning efforts. Environmental monitoring tests are done regularly, either to verify that we've cleaned properly, sanitation verification, or to find specific microbes, microbial testing. Let's talk about sanitation verification and the most common test done in the packing house. This is a nucleotide called adenosine triphosphate, or ATP. It's part of a molecule that's found in all living cells and is responsible for metabolism or growth. ATP is harmless, but when we clean our rollers, brushes, and other food contact surfaces, we want ATP gone. So we regularly test for ATP, not to find contamination, but to verify our cleaning process is successful. We learned how to test for ATP from nature. Let me show you. This packing line has just been cleaned and the crew is ready to apply sanitizer. Sanitizers are used to eliminate cross-contamination by killing potential pathogens. They work best when applied to a well-cleaned surface, and the ATP test works best when it's done before sanitizer is applied. Sanitizers can alter ATP test results. So, to get a true measurement, they first clean, then test for ATP, and finally sanitize. For testing ATP, sanitizers aren't good, but fireflies are. A firefly's light happens when a natural enzyme called luciferase comes in contact with the firefly's own ATP. The biochemical reaction emits light. This is called bioluminescence. Science has borrowed from the firefly. ATP bioluminescence is a test using the enzyme in this, an illuminameter. Like the firefly, we can mix our enzyme with samples from the processing equipment. If ATP is present, light is emitted and measured by the illuminameter. The higher the number, the more ATP there is. If the number is too high, we know we need to improve our cleaning efforts. What is that number? The answer is in your environmental monitoring plan. Your plan takes into account the brand of testing tool you have, the unique environment of your plant, and data analysis of your test results. Your monitoring plan also has guidelines for microbial testing. We test for microbes in zones 1, 2, 3, and 4. Aerobic plate count, APC, is an indicator test that looks for all microorganisms that can grow in air at 98 degrees Fahrenheit, like coliforms. These microbes won't make you sick, but they should not be present at high numbers. Like most microbial tests, APC test samples are sent to a lab where cultures are grown on plates to obtain results. High APC indicates poor sanitation, but if we find low counts, we still can't be sure there aren't illness-causing pathogens because APC can't identify them. For that, additional testing is done to find specific microbial pathogens, including pathogenic E. coli, Salmonella, and Listeria. Food contact surfaces, or Zone 1, aren't tested for foodborne pathogens, but may be sampled for indicator organisms like coliforms or generic E. coli. Let's do some test samples and learn a few common rules along the way. Here, we'll test for possible Salmonella contamination using a sterile sponge on a stick. Rule 1, our hands are washed and our gloves are always on. For pathogen tests, it's important to swab a large surface area. In this case, 12 inches by 12 inches. Identify your sample area and begin by firmly swabbing back and forth like this. Next, swab up and down using the same pressure. Finally, swab diagonally. Your environmental monitoring plan has a protocol for sampling. The rule here is to make sure you swab the same way every time. On irregular surfaces like this control panel handle, every tester swabs the entire handle. Sample sites are rotated. Testers swab different areas along this flume. They use these small swabs in order to get at hard-to-reach areas, all of them using the same technique to capture samples. Sampling consistency is critical for achieving the most accurate results. With ATP testing, the results come very quickly. Most pathogen tests, however, are sent to an approved lab. Samples are clearly labeled, including sample description, where and when the sample was taken, and any other needed information. It's now time to pack samples into a cooler with gel ice packs and sent unfrozen to the laboratory as soon as possible. The tester's last step is to record the data we've captured. We record the time and place of our sample as well as any other relevant information. This data is then entered into computers. Using data analysis to track our test results, we're able to find areas that need intensive cleaning and more regular testing. And that's all the testing we're going to do today. Well, almost. You can see that monitoring the packing house environment is as important as the efforts good people make and the quality of fruit they deliver. Let's sum up what we've learned. Environmental monitoring plans are unique for each packing house, taking into account your product, machinery, age, and climate. Environmental monitoring begins with visual inspections, keeping an eye out for opportunities to ensure food safety. ATP testing is done to verify the effectiveness of your cleaning program. Microbial testing is done to determine if there are any foodborne pathogens or indicator organisms present. Everyone on your team conducts tests the same way every time. And finally, test results are collected and analyzed to determine frequency of cleaning and areas that need special maintenance. Let's take one more test that doesn't require swabs or sponges, just your head. What is the first rule for environmental testing? Clean hands wearing clean gloves. To test for pathogens, how large an area on this surface do they need to swab? That answer's in your environmental monitoring plan. True or false? If your illuminometer reads 53 after testing for ATP, you must clean your equipment again. False. Every packing house makes their own determination on what number is too high for ATP. Let me leave you with this. Become a student of your packing house. Study the processes from packing to cleaning. Take the best, most consistent samples you can and use that information to seek and eliminate microorganisms on your line. Thanks.